Thank you very much, Rebecca, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you so much, Kate. And I'm thrilled to join you all today to talk about basically how my lab likes to misunderstand the term synthetic biology. And we'll talk about sort of synthetic biological systems built from DNA and PNA. And I think to start the just introduction, the sort of self-introduction, you're hearing from someone who actually started her career in like medical device development, product design. And so I had a career before going back to grad school and getting into microsystems, MEMS-based sensors for soft, squishy systems that interact with cells, and found my way to molecular motor engineering and now mechanical engineering as my home department at Carnegie Mellon. I'm the Ansys Career Development Associate Professor, but I'm also in biomedical engineering and electrical and computer engineering. And we actually primarily work with nucleic acids, interfacing them with both biotic and abiotic systems. So I see myself as a device developer, right? And a lot of what we're going to be, I'll be presenting are basically the tools as well as what you can create with them for synthetic biology. And I think in general, there's 4 basic stories I want to tell you, 2 longer ones, 2 shorter ones. And yeah, if you do have questions, feel free to interrupt me. Also, I've got the chat open. It's not obscuring my screen. Okay, so I'll get started. Okay, so to start, yeah, so this is a general framework for the talk. Like I'll talk about how we actually interface nucleic acids, nanotechnology with plasma membranes and applications for what we can do when we're thoughtful about how we interface with living systems like cells. Then I'm going to talk about how we move material around, which I think is an open question and major challenge in sort of synthetic cell type applications. And that's going to get into microrobotics. And I'm going to end by covering 2 different topics that are near and dear to my heart. And, one is this sort of, how to extend the field to build better nanostructures, and how do we get things like enzyme resistance by exploring the sort of xenomaterial space. Okay, so with that, so that's sort of like holistically like what the talk will be about. And I can go to the next slide. Cool. So as a mechanical engineer, sort of card-carrying engineer, I want to start by just saying that, nucleic acids are very much engineering materials. And it's a good framework, I think, to introduce people to this topic because they're really kind of oftentimes predictable in a boring in a good way. These are engineering materials. I'm showing right now a model kit that my wonderful undergraduate, Kaylee Goodwin-Schoen, engineered so that we could have 3D printable models that actually are really easy to put together, but actually hold together in your hands. And so we sometimes build these structures, and I'll get back to that later. But it also drives home DNA acts like a hard plastic, actually, compared to other materials. And so it's sort of a good framework, but it's a powerful engineering material. So like I said, the sort of 4 topic areas, we'll start with cellular interfaces. And I just want to show you kind of a smattering of examples that give you a sense for the power of working with nucleic acids, particularly anisotropic DNA nanoparticles. And then we'll go on to the microrobots for moving things, and then exploring sort of new tools to advance these fields. And I think the motivation, which I don't know if, I feel like this community is well aware, but when we misunderstand the term synthetic biology, in the DNA nanotech community, oftentimes it's trying to think about how to reconstitute functionality of certain sort of components of a living cell through a system that you build in a tube, and then you can introduce into a cellular system. And so most of these have actually been made external to cells, and then maybe be introduced to create a synthetic pore or to create a vaccine type system that can only open in the context of the cell it's targeting. You can make force application systems, and then even moving towards multi-component molecular motor ensembles like these turbines. And so there's this tremendous potential for building at the scale that the cell itself builds at. And that gives you this sort of incredible power to engineer these systems, but also to understand how biology does what it does and potentially to communicate with, right, and collaborate with living systems. Okay, so that's like the motivation. One particular example would be with DNA nanotechnology. We're trying to figure out uh, how to talk to cells, how to learn the secret handshake. And one way you could do that is just how you functionalize these systems, and another is the shape of the systems. And so you can actually engineer particles to attach to a plasma membrane, to lodge across it, to go into it and into the cytosol, or go into it and get trapped in an endosomal particle, right? And so it's like there's this idea that we can engineer these pathways. Okay. So early work that came out of my lab illustrates this really well, and so I wanted to highlight the work of Ying Liu and show here's sort of a standard way that we would interface DNA nanotechnology with a living cell. Oh, good. I'm glad that I'm glad that my misunderstanding is actually it is now standard here. Okay, fantastic. Yeah, but so if you decorate a cell membrane with single-stranded DNA, then you can hybridize nanoparticles that carry the complementary sequence, and you can show with a spherical DNA origami, like a wireframe spherical structure, that the more overhangs you have on the structure, the more likelihood there is to bind, and it has the relationship that you would expect in that, like, kind of like each additional overhang provides an independent, there are sort of independent binding probabilities at work there. So you get this nice curve that we can actually fit to a model. And what we found when we don't use a sphere, I think, is really interesting. And so here we have an experiment that I think most grad students would hate. You've got now like a sheet of paper. It's a very boring and isotropic shape, but still it's just a flat rectangle. And we don't see that behavior where it's like you can add many, many, many overhangs, but it just doesn't bind to cells well, right? And so what does that tell you? Well, it tells you that where you place your overhangs matters on non-spherical structures. And if you're thoughtful about that, then you can start trying to figure out what does work, right? And so here we have the same structure, but now you decorate the edges and the corners, and you kind of recover this nice binding curve where you can now tune its likelihood to attach to a cell. And what's really, I think, great for engineers is there are uses for both of these. So if you want an interface, and you can think of it like a lab on a chip that's at the nanometer scale, if you want to drive this to a cell surface and you want it to get to like all the cells or all the liposomes, decorate the edges and the corners. But maybe there's a use for face decoration because you can get them to attach, but you need a tether, right? So then here's in this sort of like whirlwind tour, here is an example of why that could be extraordinarily useful. In an engineered system or in potentially a disease model, you can now selectively target cells with damage to their glycocalyx. So you can use trypsin, you can also use neuraminidase, and basically degrade the glycocalyx or remove charge from it. And what we could do is show that the degradation of the glycocalyx results in enhancement of the attachment, right? Whereas previously you needed a bridge to get there. Only the damaged stuff can do it without a bridge. So it's like a really elegant way to say, okay, so if we think about how we interact with cells, you could use aptamer decoration. We've already talked about the power of cholesterol. You can use antibodies. But you could also now use the shape of your nanoparticle to regulate that behavior, right? So here we have a cholesterol attachment, but only if that glycocalyx is damaged, right? So it gives us this enhanced sort of selectivity. Sometimes you want to be really delicate. And so here we would say, okay, in this case, what I really want, if I'm concerned that my nanoparticle may actually do something adverse in a situation, we used human blood platelets as the example because you can't do much without activating them. Some people say you look at them funny and they'll activate. So what we wanted to do was figure out, can we decorate a human blood platelet without activating it? And the strategy to use would be use a shape that decorates extraordinarily well. And so we've got this rectangle, but we've got edge decoration, so we know it'll decorate well. And with this work, we can basically show that we can tune the valency of that particle to boost how many attach, but do it in a way that, once again, plot D is one of these like exciting, boring things. Can I get my, I don't know if I can get my cursor. Well, okay, the p-selectin plot is flat, which means that it doesn't cause more activation to have under these different conditions as compared to just like our controls. And so what this says is basically, if you're thoughtful about how you decorate your particle, you can get it to interact with cells, but you can do it in a way that doesn't activate them, right? Yeah, so we call this basically like sneaking up to a lion and putting a name tag on it. So you can be really delicate. And I think this allows sort of us to finally look into mechanical activation of cells, because we've got a platform that can be introduced without the activation process getting triggered. Okay, and then I think, and then I go, yeah. And because I like the superhero thing. So basically it's like, you know, making sure you're dealing with Bruce Banner and not the Hulk, which is good. Okay, and so then I wanted to also talk about how to make the introduction of these platforms practical and really useful. And so this was work from a team of students in my lab, and we were exploring the different ways that you could interact with cells. And so one way is you can put these cholesterol anchors in the membrane, and then at a secondary step, introduce your origami. Another thing you can do is you can actually feed the cells an azide sugar, and then they'll actually build a tag that you can link DNA to in their glycocalyx, right? So, so that's another really distinct way. It's kind of like attaching to the top of the trees versus the ground floor in a forest. And then there's a third way you can do this, which is to put the cholesterol directly on the origami. And there were some great innovations in 2019 that made it possible to do this without actually leading to aggregation of the particles. So there's many ways you can do this. Doing it in one step would be incredibly preferable. And it would be great to be able to do it in a way that got you dense decoration. So sort of understanding those rules has been something we've been trying to understand. So here is the plot that I would want you to take away. So these are all sort of normalized to their initial decoration capacity. But if you look at an undecorated rectangle, a densely decorated but only face decorated rectangle, and then an edge decorated rectangle with only 4 overhangs, you can get sort of massive improvements in your ability to label cells if you play with the position of those overhangs, and you can get there far more efficiently than you could with even having more overhangs on the rectangle. And so we see these big advances, particularly for the rectangle, but we also see improvement for the nanorod. For the sphere, it's more that, you know, there isn't really like an edge in a corner. So the more the better in terms of labeling, and they actually decorate really well. So we're sort of normalizing to themselves is a little misleading. The spheres do wonderfully, but they decorate everything. And you don't see much improvement with additional overhangs. Okay, so we're sort of learning these rules and learning that not only did it have to do with labeling, but also uptake was fairly proportional to how well you could attach to cells. So if we're trying to learn the game of how to get something onto a cell and how to get something into a cell, it's that anisotropy as well as the placement of overhangs. And then the third thing would be, how do we do this easily, right? And so for these studies, we basically compared non-specific interactions or non-targeted interactions with our traditional 2-step cholesterol, and then this modified 1-step where the cholesterol is put directly on the origami. And this was based on an approach that came out of Ulrich Kaiser's lab. And I saw that Ulrich Kaiser is speaking in a few weeks, so that's going to be fantastic. And so this was published in 2019. And basically, we extrapolated this method to then show that indeed you can get really good labeling, even without doing both the separate overhang or initiator labeling followed by interaction or introduction of the origami. And kind of what that means to everybody should be this. The fact that the light green is more than half the height of the dark green means that a one-step labeling method is actually fantastic. It's good. It's not as good as two-step, but it's really good. And then you could imagine starting to deliver these potentially in vivo, but in environments where you don't want to flood a system with an initiator, right? You can just have one particle. Yeah. So anyway, so that's sort of how do we stick a particle to a cell and what happens. And then I think for the only thing that might be really going to the traditional definition of synthetic biology is what's been really interesting in the nucleic acid field is you can wrap up a gene and make that your nanoparticle. And our lab is not ready to publish on that work yet, but what I can show you is that even with something as simple as cholesterol anchoring, the confocal images suggest these particles are getting to the nucleus. Right? So it may be that there's sort of a new field of how to get nanoparticles to the nucleus with cargo that includes gene material. Okay. So then one other thing that it's really interesting about labeling cells with nucleic acid nanotechnology is that you can be the opposite of delicate, right? So you can greatly modulate cell behavior. And in our cell armor paper, Weitao Wang explored what happens when you use that glycocalyx anchoring method to enable the binding of lots and lots of nanorods on a cell surface. And then he introduced another species of nanorod that could cross-link that first shell. And so what that means is that, we're basically turning the cells into Iron Man. Right, and trying to explore what are the benefits of effectively ruggedizing a cell? Because what you see here is when you add rod A and rod B, you actually do get aggregation. It forms the secondary layer, but it would be sort of geometrically limited. And in this paper, he then went to go explore. Okay, and so the first thing you can see is depending on how you set up this nanoshell, which basically these numbers mean how many overhang binding sites there are between the two different species of rods, you can actually carefully modulate the effective elastic modulus of the cells, right? And so we can more than triple their mechanical stiffness. And there are other things, right? And so in terms of membrane fluidity, you can see a reduction in mobile fraction, right? And you can see an increase in this time constant, right? So we're actually changing how the lipids are moving around on the membrane, constraining them with this shell. And then functionally, that's really, really useful if you start subjecting the cells to stress tests. And so here we have a demonstration where we basically are torturing these poor cells and we're putting them in lower and lower salt concentration solutions to swell them. And what you can see in the bottom row, but also the blue condition here, is that you're preserving that, that cell size for the case with the two-part cell shell, right? And then you also kind of see this neat benefit just from rod A, which we weren't expecting, right? So these are, but you basically keep the cell from exploding in the zero salt condition if you've got the shell. So this is potentially really, really useful and useful against a range of stressors. And so we used osmotic swelling, centrifugation, and high shear injection in order to see if these shells could increase the survival of the cells. And indeed, we saw a boost from just rod A, but we saw a much greater improvement in survival for having both rod A and rod B. So now we have a potential way to strengthen an arbitrary cell, and you could imagine targeting this to certain cells that have certain properties for temporary protection. Okay, so that's my first story. And now I'm going to talk about robots. Okay, and so I think that the way that I would explain this is we want to interface with living systems and manipulate them, understand them. And one of the key parts of that is this communication, this movement of materials and information, right? And so the way that I would typically set this up is to say, you know, if you wanted to perform targeted drug delivery or micromanipulation or microsurgical activity, or even just kind of like circulate liquid in a microfluidic device, having something that you can control using an external magnetic field that manipulates matter at the micron scale, that's extremely useful. And if you were in the sort of synthetic cell universe, you would want to move material from one place to the other, potentially move cells from one place to another, without disrupting non-magnetic systems, right? And what I can show is we have a paradigm for a magnetic microswimmer that's really simple and potentially extremely useful. So for this, as engineers, we look at sort of the materials we can build with at the micro and nano scale. They're increasingly complex and really useful, a sort of DNA origami type nanoparticles. Cells themselves are micron scale. There's a range of colloidal particles, and then also sort of more simple particles that just have like a polystyrene sphere or one with a magnetic coating that we can choose from. And that's what we started with for these studies. Our robots also have flexible filamentous components. And so we could look to biofilaments, right? Like microtubules, actin, but also peptides like that have alpha helices, for example. And then we can also make structures with tunable mechanics from things like DNA nanotubes. All right. And so here is the kind of swimmer I'm talking about, and get ready. It's not going to move fast. All right. Oh, it didn't move at all. Okay, that's very slow. All right, so this is for us, our microswimmer. And I want to show you the benefit of a system like this is one of these particles is magnetic. And if they aren't connected and you apply a constant magnitude oscillating field that rotates, the particles will just experience, the magnetic ones experience a torque. So they'll just rotate back and forth. And the non-magnetic ones won't experience a force. But if you've formed a robot that has two or more components, then those systems can actually move. Right? So if you can logically control the assembly and disassembly of these swimmers, you can make robots that move around on demand under the control of a magnetic field. Okay. And what we've been trying to figure out in our lab is, how should we make these? Okay, so first and foremost for this one, here's where we're going to draw on the sort of top-down approaches using lithographic approaches so that we can actually template the placement of these particles and use those templates to filter for the particle sizes that we want. So we can actually get a more monodisperse outcome than you would expect from the input materials, right? And we use DNA to actually link them together, right? And so you can see our little duck-like swimmers where you've got the magnetic particle, the non-magnetic particle, and you can kind of see a collar there. And this is just this sort of lots of nanotubes linked together, holding them together. Okay. And so with something as simple as this, we've been trying to understand how do the different relative geometries, magnetizations, and actuation frequencies impact their swimming properties. And then I actually had a question, if I missed that, if the spheres are empty inside. And so what I would say, so I'll back up and say, right now, we use polystyrene spheres, but at present, we're using streptavidin-coated spheres. So you could link like a drug of interest to the surface at present. And the models that we would think about going forward would be alternate particles that could be loaded more densely. So if you use like a nucleic acid-based particle, you could load drug by intercalation, or if there's something that you can modify to attach it to DNA, you could do that. But something like a vesicle, you know, like a lipid nanoparticle could very well be the cargo. So as long as it's at the micron scale and it holds together and we can attach to it, I think the sky's the limit. And we're interested to explore that. So I think this is actually a really good segue to talk about why this is hard. Okay, so one of the things that you see when people make micro robots is they'll oftentimes just show you one as it's swimming, and I totally guilty as charged, right? And the reason that they'll show you one is that if you made 50, they will actually usually swim in all different directions, right? And they may swim at lots and lots of different velocities. And they may also get stuck to the surface. And so we have this big challenge of trying to understand, given that tolerances get relatively larger as your dimensions get smaller, we're building a really heterogeneous set of robots, right? And we also aren't controlling the sort of direction of magnetization of these particles. So we have trouble, and we're still working on controlling the direction they swim. But just due to the heterogeneity, it's hard to actually engineer and explore to optimize geometry locomotion properties. And so then that's what the next slide, I think, is about. So what we did was we stepped back and we said, okay, we can build something at the millimeter scale. It's huge for us, right? But we can do it. And so here we can use 2-photon polymerization, and we can start making mimics of our micron scale swimmers. And then if we swim them in a more viscous solution, we can get the same Reynolds number. We can mimic the same swimming parameters. And so this is what we did. We basically finally had a platform where we could say, we can control the geometry and make a highly replicable geometry. And we can then explore how do these things swim when you have a cylinder, when you have spheres, when the sphere relative sizes are different, when the cylinder aspect ratios are different. All right, so this was the work of Emma Benjaminson and Taryn Imamura. And with this, we found this really interesting. This is our little yellow submarine project, where basically you could see that depending on K, so depending on the stiffness of our connector, we saw changes in the maximum velocity of our system, right? And so there is, at least in this regime, a softer spring gives you better locomotion, which makes sense because you have to break the scallop theorem. And if you've got a rigid object at the micron scale and low Reynolds number, in order to have a non-reciprocating gate, there has to be kind of a lag between the two pieces moving. Otherwise, you can't locomote. You actually can't move in low Reynolds world. So weak spring is useful, but then also we could start exploring what the relative role of things like aspect ratio are. And you can see that these sort of maximum frequency or the maximum velocities occur at higher frequency, right? For our larger aspect ratio. And so this gives us guidance on how to design. So we kind of in parallel have now engineering at the millimeter scale and the micron scale, but the millimeter scale can inform this. We're also doing work in computation, but this in and of itself is complex because you've got elastic components and you've got fluid structure interactions, right? And so this is sort of one of these new challenges for the soft squishy robot simulation. Most of our computational fluid dynamics modeling is not set up to do this well. So yeah, anyway, okay. And then so with this story, basically we began to learn how should we build our robots? And then we also addressed the yield of these systems so that we can make hundreds and hundreds and hundreds of them, or thousands, depending on the scale of our template. And so we have a recent paper out where Taryn she tuned the surface energy of her templating material. So she actually ended up using polycarbonate instead of PDMS for her material. And in that case, she could show that going from like basically non-existent pocketing yield, like non-zero, but very low for PDMS, she could get a really, really, really high yield pocketing behavior. And that now is able to explore what happens when you make ever more complicated templates and you want to get multiple pocketing events to happen, right, to build a structure. And so here we have structures that have, 4, and then I think 10, and as many as like 18 different components, right? And we can see that we can actually get them well templated. Okay, so the future of these robots is that we now get to think about body plans and how that also interacts with function. Okay. All right, so the big takeaway from this should be, I think, that we have the ability to interact with matter at the micron scale with sort of unprecedented control, and that nucleic acids provide an extremely powerful way to do that. But when we think about nucleic acid nanotechnology, I want you to think about the wireframe spheres and the sheets of paper, that I showed you. So there's this promise, this tremendous promise that you can build anything, right? And so why on earth would we build the most simple, most sort of boring shape possible? And the reason for that most of the time is that it's just practically extremely hard to build sort of unusual geometries. And that's a shame because it seems that the particle properties, like number of pointy corners, flexibility, stiffness, like all that's extraordinarily important to the particle itself. And so what we've been thinking about in our group is, how do we push the field forward so we can do automated design and design ultimately for function? And so I can show you a few examples of what we've worked on in the past few years. And so this is the work of Tito Babatunde in collaboration with John Kagan. And here we have a generative design process. So Tito actually encoded like a formal language to describe honeycomb DNA origami design. And then she let the computer do the routing to get these unusual shapes, which would be extraordinarily burdensome to make by hand, right? And this is just sort of the starting point to show, okay, well, we can fill up a shape. Great. But you can start doing really interesting things that a human couldn't do. So you might say, like, well, I have a fixed amount of material to build my particle with, but I want to make it fit into this shape. So why don't you just use up all the material I have, and then we'll have a hole in the center, right? Well, that's a very reasonable thing to do, and the software can now do this for you to give you hollow nanoparticles. You can coat the inside or the outside of the particle or the template. But this is this idea that a human would find this extremely challenging. And our goal is to get to the point where we're saying we want properties of nanoparticles, and then the software will generate hundreds to thousands that we can then pick from and computationally compare. Another useful and sort of parallel direction that AJ Vetterini in our group advanced is basically to do the wireframe version of this. So you can, in an automated way, generate wireframe DNA origami. There are sort of multiple really powerful tools like vHelix and the Athena suite that will let you take a triangular mesh and convert that to DNA origami. And so what AJ said was, okay, I'm going to make those meshes, and I'm going to figure out ways to do it in ways that can help people. So for the sort of user-defined inputs, you might want to work with some crystalline lattice. So you might want each nanoparticle to interact with its neighbors in certain ways, for example. So you could set these sort of green points to say where we definitely want connection points, and therefore design for some sort of lattice. And then you can also say, but I don't want material where I know I'm going to have my nanoparticle or my peptide or my protein or whatever I'm going to encapsulate in my particle. And so you can actually say, this is hands off, like nothing can go in this region, and then run an optimization. And this can be either like kind of single objective, like explore porosity, maximize it or minimize it. But it could also be multiple objectives, right? Which is the way that humans design. And then this system will give you a population of nanoparticles that allow you to explore the frontier, right? So, and sort of understand how these trade-offs work. So this tool is now published as well, and what we're hoping to enable users to make ever more interesting and useful sets of nanoparticles in a generative way. Okay, so that's the short the first of my short stories. And then I think the last piece of this has to do with DNA mimics. And forgive the use of like Copilot or whatever, but my I had all these superhero pictures, and so my colleague generated this for me, which is that's the DNA mimic, in case you're wondering. And for those of you who aren't familiar with peptide nucleic acid, PNA, this is my summary slide that kind of tells you what you need to know. All models are wrong, many are useful. PNA doesn't exactly have the same helical properties of DNA, but if you can start by thinking of it as a less twisted nucleobase bearing information polymer, right? So it doesn't have the highly electronegative backbone of DNA. So without that sugar phosphate backbone, it's actually uncharged, and it's got an aminoethyl glycine backbone instead. But it's got sort of similar axial rise, and it's less twisted. And it has really fascinating properties because of that lack of charge. Once it binds really strongly. And you can get strong binding, like they reported recognition domains as short as 5 bases. And it doesn't or it's more conformationally constrained, and so it doesn't actually bind mismatches really well. So you have to get the right sequence. It's better at binding the right sequence. So for an engineer, this is extremely exciting just in terms of its high affinity and its mismatch intolerance. But it also is not recognized by nucleases and proteases. So it's not a substrate for either. So now this is a material that's very much like a Trojan horse, and this could be really useful if you wanted to have a sensor that could survive in or out of the lab and persist for a while, right? So I think that would be really useful. Most of what we do with DNA nanotech, we sort of use it within a few days and then we assume it's going to degrade. But this would actually change the calculus there a great deal. Okay. It's also used for turning on and off gene expression, so it's really useful in a sort of genetic modulation. Okay, so that's my big pitch for PNA is really useful, but we don't know a whole lot about how to build with it. And so here's where I want to come back to some of this 3D printing stuff. So my lab, made this tool to put together 3D printed models of the molecular systems of interest. And so here's a DNA duplex, PNA DNA duplex, and PNA PNA duplex, where you can basically see the compatibility between the white DNA and the purple PNA. But it has an altered helicity in addition to sort of an altered helical geometry, which may make things complex. And so one of the things that we've been actually relying on is we make models of the PNA systems to verify that the structural motifs that we're engineering can work. And so this is where I would say everything in here is published with the exception of this work, where we've now made automated tools to help people make 3D printable molecular models. Because we think it's such an incredibly useful teaching tool, but also because it's essential for understanding how these systems actually come together, particularly if you're working with something like PNA, which is going to be constrained in ways you didn't expect, right? And so this is actually really useful from a design perspective. And we're looking for beta testers for this tool, and so you can reach out to me if you want to 3D print your nucleic acids, but also kind of like any small molecular structure of interest, we'd be open to talking about. Okay. But I can show you our first stab at PNA engineering basically just said, okay, so it's less twisted than DNA. Let's make filaments that take that into account. They've got shorter interaction domains because it binds really strongly, right? And we just want to form, let's say, 3 helix bundle, right? And so with this structure, we actually found that we could form filaments, but they didn't follow the rules of DNA. We were expecting filaments on the micron scale, but we were expecting them to be like 8 nanometers across. And we found a wide variability here, and you could actually see the sort of negative stain in the grooves here, showing that for this TEM image, we think we're looking at a bundle of these that have come together. I think Helga, this is the question. Oh, I can maybe I'll go to the swimmer question later, but yeah, Molecule Crafter is built on the, backing up a bit. The software tool is kind of an add-on in a student accessible, well, open, it's open as in like everyone can use it for free, but it's a web-based CAD tool, right? And so ultimately it's a tool where nobody needs to install it and you can use it in the browser, right? But it's not fully open source, but it's within Onshape. And then the question is, can you form PNA origami? And so what I would say is our lab is actually now engaged heavily. We have an ongoing grant, and I didn't want to show unpublished data, but essentially you can make PNA DNA hybrids, right? And so if you bind PNA to DNA or PNA to RNA, you get an intermediate helicity, sort of an intermediate conformation. And you also make it hard to recognize that stretch of DNA or RNA by nucleases, which is useful. But you definitely can integrate PNA into a DNA origami. It does something called strand invasion because it binds so strongly. It can actually sort of reconfigure your origami, and even without a toehold, get in there and replace part of a DNA from a duplex. It also does triplex binding, right? So it raises all these interesting questions about how should we be moving towards something like PNA origami. And at present, in this paper, basically, we titrated in increasing amounts of DNA into the structures. But our goal for a lot of this work was to make entirely PNA structures. And the biggest issues that we would see is, as this slide highlights, you get behavior that's not like purely DNA. Without that charge, you start getting the sort of peptide-like behavior you might expect where you get a hydrophobic core, and things sort of tend to crash together. So we have to modulate solubility a lot. And there are multiple ways. You can put terminal lysines on things. You can use these backbone modifications. And so that's one of the big challenges for PNA origami is figuring out how should they bind. And then the next, and that's kind of where we are for learning the motifs. The other thing we need to figure out is how do we learn how to form them? Because we need structures that form with high yield and ideally structures that form rapidly. So like, one of our first few papers on PNA, we're exploring isothermal manufacturing. Yeah. And then, so PNA, and then that's a great question. Like, how do you store it? You can store it in the fridge. You can store it in the freezer. We have accidentally stored it on the benchtop for 2 years, and it still worked fine. And so I think in general, it's less finicky because it's far more stable. Yeah, that's a great question. And can it be used for information storage? I mean, I tend to think it could be used for all manner of things. You would just have to figure out how to work with a fixed set of PNAs. So the difference between PNA and DNA is that you may only ever have access to dozens of unique sequences. We have a project where we're attempting to get access to hundreds of unique sequences, but that's huge. And so if you want to store information, I think it would be fascinating to use that for storage, but it would have to be sort of an information sparse approach. Yeah, that's a great question, Mohammed. Okay, but we can now make these filaments robustly, and we've been focusing much of our efforts on making them soluble, and then also currently learning some of the rules about how different modifications of the PNA actually affect its ability to form nanostructures. And I could say that's what I could report on next time. Yeah. Here's an example of regulating the assembly of a PNA system with the presence of a DNA template, right? So you can either promote long filamentous growth if you have a small amount of template that nucleates the growth, or if you flood the area with lots of template, you can basically use up all of the PNA and form lots and lots and lots of little particles. So we can actually see that there's a tremendous ability to sort of regulate these systems with PNA. And I think the cell permeability question, so basically in comparing PNA and DNA, Ranjan has asked, how cell permeable is it? And that usually comes down to charge, right? So if you put a positive charge on PNA, you can get it out of the circulation and get it into cells. But I think that's one of the big challenges that folks have dealt with for the gene delivery applications, because I think the other thing is just that solubility is generally an issue. So you want solubility, but also if you can promote its interaction with membranes, that's really useful. Okay. Yeah, and so I think with that, we can open it up to more questions. So in summary, today, what I showed you was that nucleic acid nanotechnology has an important role to play in interacting with and manipulating membrane systems like living cells, synthetic cell systems. And if you're working at that scale, it's also important, I have my pictures flipped, it's going to be important to move material around. And I think we've been positing that magnetic fields can help get the energy into the system in order to have micro robots drive around cargo and move things around in directed ways that don't disrupt the larger system. So at the micro scale for manipulation, we think these slow swimming micro robots have tremendous potential. And then in order to push the field forward, there are two big things we need, and it's that we need tools that allow us to be more thoughtful about how we design nanoparticles, which can be really cumbersome and highly geometrically limited at present. And then finally, there are these other materials we want to explore, and PNA we think is particularly interesting, and it would be great to engage with folks in this community to talk about other possible sort of xenonucleic acid approaches or modified DNA type approaches, because some of these generative tools that we've been developing are completely agnostic to the helicity of the material, you know, and so we are at a place where we'll be able to design nanoparticles to be made of whatever material we can generate. Okay, yeah. And I think with that, I want to give a pitch that we're also doing some cloud work, automation work, trying to figure out what the next generation of production of these particles looks like as we're trying to scale up and improve quality. And then I wanted to thank all the wonderful students in my lab, our collaborators, our funding sources, and all of our facilities and administration here at CMU. So yeah, thanks. Happy to answer questions.
Thank you so much. This was awesome. I think there are a few more chat questions that
oh yeah, I think there was one.
I think Nate's is the first one that
I missed.
Yeah,
at
11:32.
So these are ferromagnetic, and it's possible to do this with super paramagnetic as well. I think one of the things that you work with is you need to be able to generate enough force, right? And so the ferromagnetic for us, for the torque, has like never caused issue, but I think that both could potentially work. One of the really most simple things as a note that would help us figure out what it actually takes, you know, what the actual requirements for these particles would be something like optical trap studies, so that we could actually see what it takes to hold our particle and keep it from, or I guess just like we don't know how hard how do I say this? We're in a strange situation because actually what we would like to do is see if we can remagnetize our particles. But we actually don't know that we can do that because they just rotate in place. So it's kind of like, it's always hard to know if you remagnetized or if you just distorted your particle structure, right? And so if there were wonderful ways to temporarily immobilize a magnet or a magnetic particle and then measure the forces that we're applying or the torques that we're applying, that would be really useful for us. And so we're at a point where I think there's a lot to be learned with these building materials, but yeah, the chemistry is involved. I think as you get smaller, oftentimes these things are super paramagnetic, but we use spherotech spheres, which are described as being ferromagnetic. Yeah. And we don't see any issues with aggregation, although I think it can be really useful to have a super paramagnetic system that doesn't have the magnetic properties when the field's off, right? And then you would basically tune the particles if they're ferromagnetic so that you use a low enough density or you coat the surface so that they're less likely to like crash together. But these are really interesting concerns. Are there any other ones that I missed?
Did you answer the one about the towing a cargo? I forgot.
Oh, yeah. Okay. So yeah, so I mean, at the micro and nano scales, the ability to tow a cargo is, in terms of the energy it would take to move it, I don't think would be an issue at all, right? You could imagine, you know, molecular motors towing like vesicles. The real question, though, would be how does it change your swimming, right? Because if the whole way that you locomote is essentially that you generate differences in drag on two bodies, if you have a third body that rivals the size of those first two, that presence of that third body may actually affect your swimming speed, right? So they would have to be studied, but we actually have a platform to do that. The other thing that's really interesting, though, about that could be you've been planning to drop certain cargoes, and as an indicator that you drop the cargo, your swimming capability would change, right? Like once you dropped it, maybe the swimmer would like accelerate off or you know, just or speed up across the.
That would be cool to
So I
see,
think there's a lot,
honestly.
yeah. And these are the kinds of things we're trying to simulate now and actually build at the millimeter scale. So yeah. And then so I see Prathamesh has asked, what applications do you envision for microrobots? And yeah, I think most people will typically like show them in a blood vessel, you know, and I think that for delivery in tissue, being able to navigate the circulation is, you know, bacteria do this even without being able to swim faster than blood flow, you know, so I think that there are actually reasons to be optimistic that that's actually, you know, very, very plausible. Some of our first applications, we were talking about targeting to low flow environments. You can imagine like the viscous media in the eye, where you need very, very careful delivery. Yeah, but I think they need not be limited to the biological realm. I think that more broadly, just the question of reconstituting machinery to do work at the micron scale, we either have to build a network of like transport microtubules, you know, and figure out how to actually use these highways and between our I don't know, critical points on our landscape. Or we can also think about ways that are much more kind of synthetic to go and move material around. So from a manufacturing perspective, I think that there's a lot there at the micron scale. And I think also when you imagine that a nanoparticle is oftentimes hard to watch if you have just a standard light microscope, But if your microswimmer is like representing an intact assembly of multiple systems at the nanoscale, you can also read that the assembly and disassembly of your robot to tell you kind of the state of the nanosystem within it. So I'm hoping that we can get insight into nanosystems and also move material around outside the body with these microswimmers.
I'm really excited. I just want to say, I would love to see some of the applications, especially on the interface between the synthetic and the more biological. That's where that's the real, groundbreaking place for new things that neither nature or the traditional robots can do.
Exactly. And then we have models kind of, if we can leverage our insight from the macro scale and insights from these millimeter scale experiments, you can build what nature never really, I don't know, settled on.
Yes.
Yeah.
That's the best part for me, is do something that nature never bothered doing. That's the fun part.
Yeah.
All right. If there are no more questions, I want to say thank you so much again for a fantastic talk.
Thank you so much for having me. This was really fun. It was wonderful questions.
Why is there a dinosaur on your campus?
So that's Dippy, and this is one of the Carnegie museums and Carnegie libraries. And I think that the donor also funded some exploration where they discovered this dinosaur, I want to say. But also we just kind of put scarves on him. And so if you're in Pittsburgh, you can tell the season by what Dippy's wearing.
Oh,
it's kind of our mascot. Yeah.
That makes sense. We only have a giant rodent statue on campus. Not as cool as a dinosaur. All right. Thanks so much. And thanks, everyone. Have a great rest of your week. Bye.
Bye-bye.